and often a response to genetic is, well, nobody in my family had this, it can't be genetic. I think there's a lot of genuine misunderstanding. I think, at essence, when we say something is genetic, we are saying that if you look at a gene or DNA, which is the way we look at genes, picking up some change, and we now call that a variant rather than a mutation, that is altering the way in which that gene is working, and that is what has produced, in this case, cardiomyopathy.
Welcome to Cardiomyopathy Conversations, the podcast where we talk to medical experts and patients about all things cardiomyopathy, brought to you by Cardiomyopathy UK, the UK's foremost charity for heart muscle disease. Don't forget, if you enjoy this episode, to hit the like and subscribe button so we can notify you of every new episode. If you want to find out more about cardiomyopathy, check out our YouTube channel at youtube.com/Cardiomyopathy UK, or take a look at our website at www.cardiomyopathy.org. So welcome everybody to this next episode of the Cardiomyopathy Conversations podcast. It's Libby here, and I am delighted today to be joined for this episode by Professor Ruth Newbury-Ecob, who is a consultant clinical geneticist. She is an honorary professor at the University of Bristol, and she is the genetics lead for the Inherited Cardiac Conditions Service in Bristol. Ruth, it's a pleasure to have you on the podcast. Thank you very much for joining us today.
Thank you for inviting me.
We were just saying genetics is a complex topic, and I think the idea of this podcast is just to sort of clarify a few things, really. So if we think about the language of genetics, when we say that a cardiomyopathy is genetic, what is it that we mean by that? So what is the meaning of the word genetic?
I think you've started with a really good question, because I think when people hear the word genetic, they immediately assume inherited, and the two are not the same. So actually by genetic, we mean that there is some role or function of the genes in that particular condition, but that doesn't necessarily mean inherited. Inherited would mean that there was a genetic variant or mutation that was being passed on from generation to generation. So I think some conditions will be inherited, and that not all genetic conditions are inherited.
You see, right there, we're not even 5 minutes in, and I feel like I need to get a pen and paper out.
Yeah, it's very complicated. And the word genetic triggers so many thoughts and feelings for people that does go back into their DNA and their ancestry, and often a response to genetic is, well, nobody in my family had this, it can't be genetic. I think there's a lot of genuine misunderstanding. I think, at essence, when we say something is genetic, we are saying that if you look at a gene or DNA, which is the way we look at genes, we're picking up some change, and we now call that a variant rather than a mutation, that is altering the way in which that gene is working, and that is what has produced, in this case, cardiomyopathy, as opposed to an environmental factor or an infection. So something that is non-genetic. A little bit complicated there, because of course they work together.
So if I, for example, am born with, let's say, green eyes, I've inherited green eyes from one or other of my parents. Is that a genetic thing? Is it an inherited thing? Is it a trait? How would we describe that?
All of the
above.
All of the above. So we pass on to our children a set of genes, and the basis is that each parent passes on half of their genes and the other half comes from the other parent. And within that are the genetic variants that produce what sometimes are called traits. Eye colour is a good example of that. Height is another good example. But they will also pass on any variants in their DNA, changes in the gene that lead to diseases. And the difference is whether or not those genes are harmful when inherited in a single copy, or whether these are changes that predispose to a condition, but then other factors have to come into play for them to show themselves. So a trait, often we refer to perhaps things that are more variants of normal development and normal health and well-being, and we talk about variants and inherited disorders where we're talking about diseases and disorders that affect health more significantly.
Okay. And we have thousands and thousands of genes, don't we?
So yeah, somewhere 22, 23, 24,000 different genes, and the letters of the DNA code making up those genes is 3 billion, so that's 3,000 million letters.
What you look for when you do a genetic test is a spelling mistake.
Yep.
So it's needle in a haystack.
Needle in a haystack. So there are 4 letters make up the genetic code, repeated in different ways over and over again.
Who decided that? Where did that come from? It feels quite sort of untangible, doesn't it, to the lay mind to think, your genetic code is made up of 4 letters. What does that mean,
Yeah, so it isn't really made up of 4 letters. It's made up of bases, which are the chemicals, and those are the names of the chemicals. But essentially, there are 4 different chemicals in the DNA structure that forms the template or the code. And then there are processes by which that is copied and translated and builds the amino acids and the proteins. But the core code, there are 4 different bases, A, T, C, and G, and they are repeated over and over again in different combinations.
So cardiomyopathies, they're often classified as genetic or non-genetic. And then on one level, that sort of seems fairly self-explanatory. If on testing, then that you either find a gene or you don't find a gene. If you don't find a gene, does that mean that the cardiomyopathy is not genetic?
No, very much not the case. So I think what you're alluding to is that there's a range. There's a huge range. Cardiomyopathy is not a single condition. Many of us feel now this is a final end pathway process that has many different causes. Some genetic, very strongly genetic, some very strongly environmental or infectious, and some will be combinations of that. So wholly genetic or wholly non-genetic would be the extremes, but most cardiomyopathy fits somewhere in between that. So you may have a genetic predisposition, but then something else happens before you show that cardiomyopathy, and we have at the moment very limited understanding of that. So if somebody undergoes testing, there are a number of outcomes. So we take a DNA sample, we send it to the lab, and we ask them to look at a number of genes we know cause cardiomyopathy. At the moment, testing would only look at a number of genes that cause cardiomyopathy. Of course, whole genome sequencing, which is the gold standard, would look at every single bit of the genome. When the scientists analyse the data from sequencing that DNA code, they may pick up changes that are different to what is expected as normal. So we have a hypothetical normal DNA code, and they're looking for differences, and they're called variants. And some of those very clearly can be seen by the scientists to alter the way in which the gene works and therefore be predicted to cause cardiomyopathy. And that would come back to you as a result that says, yes, you have a hereditary cardiomyopathy caused by a variant in MYH7.
How does that happen, Ruth? I mean, I think it was back in the 1980s that the first gene causing cardiomyopathy was identified. How does a gene become known to be a gene of significance in a particular disease?
So a lot of the early work that led to identification of the genes used samples from families and techniques that really quite antiquated now, called DNA linkage, where scientists looked at DNA markers, so they label bits of the genetic code, and look at the way that that's tracking in families, in people who have cardiomyopathy as opposed to people that don't, and then using actually statistical probability to say, it's likely that that bit of the chromosome 4 or whatever is tracking, and then do some gene hunting.
So you identify the gene that you think might be causative, but then you almost have to see how that plays out.
So there are a number of ways of evaluating whether those gene variants are really significant. More and more now we use computers and predictions using various programs, but you can do biological studies and actually look at the effect on the protein, look at the effect on a muscle cell. So the first part of the process, identifying the variants, which now is done on great scale and will yield a lot of potential variants, requires processing with computer programs to narrow it down to, as you say, that one in a haystack, which might actually be the key finding. So if at the end of that testing you don't have an answer, it does not mean it's not there. It just means it hasn't been found. And we have many families with very clearly genetic hereditary cardiomyopathy who as yet are gene unknown. And the researchers, that's the value of genetic research, cardiac research, is it that there are new genes that we haven't discovered yet, or is it that our ability to filter that complex, vast amount of data from the DNA code means that there are changes there, we're just not seeing them?
Or is it that the cardiomyopathy is actually not genetic?
Yeah. So that's the other option.
And I think that's quite a tricky thing from a patient perspective. You know, have I not got a gene, or have they not found a gene just because they haven't found the gene that has caused it in me? Or in fact, do I have a cardiomyopathy that is caused by something else that is not of genetic cause?
I think that most people feel that at the very least there is likely to be some difference within your genetics that makes you more susceptible. So if we look at hypertension, diabetes, people with hypertension, diabetes don't have a single gene cause for that, very rarely. What they have is a genetic susceptibility, and that is part of their often familial DNA variation. And that may well be the case. It may not be a single variant, it might be multiple variants, and it may be that you've got some from one parent who has a cardiomyopathy, but something from your other parent that is protective, and so you don't develop the disease. There's no question that cardiomyopathy can and is caused by other factors, but I think even where you see infection, viral myocarditis, in some cases we know there is a genetic predisposition there as well.
So historically we would think that there was a single gene linked to a cardiomyopathy, whereas now clinical thinking has changed slightly in that actually we're not looking for one single spelling mistake, we're looking for polygenic causes of disease, and that could be the predisposition of your genetic makeup. It could be 2 or 3, 10 or 20. I don't know, could it be hundreds of genes that work together to give you a susceptibility? Could be.
Yes, I mean, if you start to see this, particularly perhaps milder forms that develop later in age, may be very polygenic. So one of the things we do know is that cardiomyopathy presenting at a younger age is more likely to be genetic. But cardiomyopathy presenting with heart failure in later years is perhaps more likely to be polygenic and also to have those environmental influences. And more people are having testing, and that is all informing this debate. So when people undergo whole genome sequencing, say for example for cancer, we may pick up a cardiac gene abnormality. We then do the test, we find that that individual is actually currently very well.
What do you do with that information?
Share it. So anybody undergoing whole genome sequencing within the NHS consents for certain additional testing to be undertaken, and that information will be fed back to them. So for example, children undergoing whole genome sequencing for rare genetic disorders, there are optional extras, and it's very important to collect that data and evaluate it so that the right information is given. Is it different if we pick up a gene change when we're not looking for it, as opposed to in somebody who has the disease where we're looking for it? Is that information different? And I think we think it probably is, but nonetheless helpful to know about it, because you can undergo screening, and if you do have early changes, you know about it, and that feeds into your treatment. Sometimes, I think we have to accept that our knowledge still is catching up, and we may be sharing results with families where we're saying we've detected this variant, but we're not absolutely certain what it means. We're not sure if it's
the dreaded variant of unknown significance.
Variant of uncertain.
So you have a really nice alternative name for that, don't you?
Very unhelpful statement. Fortunately, huge amounts of work has been undertaken globally to rectify that problem. Guidelines have been published by the American Society of Medical Genetics, are now universally employed, and they set thresholds for reporting that mean that there's very much less reporting of variants of uncertain significance. Also, the understanding of how variants work is all increasing exponentially. We have an ICC MDT where we discuss cases, and sometimes variants were identified 10 years ago, and we're asking the lab now to reevaluate, and often those are now downgraded to VUSs, and we go back to the drawing board.
Yeah, that's an interesting point though, because I was thinking the idea of going back to a database of what genes people have. When you discover a new gene being causative for a cardiomyopathy, what is the process then, or is there a process for actually going back to your list of patients and saying, well, actually, do we need to retest all of these people? I guess in a way, whole genome sequencing makes that an awful lot easier, because you don't have to retest. You've got all the genetic information there. Talk to us a little bit about whole genome sequencing, because it's the gold standard. What is it, and how is it different to sort of gene panel testing?
So when you do traditionally cardiomyopathy gene testing, well, originally it was done on single genes. So you pick your gene, look at that. That was negative, go on to the next one. Then they were put into panels that varied, maybe 10 genes, increasing upwards to, I think at one point we had a panel that's about 100 different genes on a pediatric cardiomyopathy panel.
Wow.
The next step up in terms of testing is that you are still looking at panels, but in the process of producing the data, you have sequenced across the entire genome. So if you imagine it a bit like a library, you've got the whole library there, but what you're going to do is you're going to select certain books and look in those books. But the library's all there, and if you want to come back to it 5 years later, you can do that. You can come back and look at the cardiomyopathy genes, but the interest for the NHS and public health going forward is that your data is there, and you can come back and look at diabetes and cancer and whatever else you want to look at. So the sequencing has been done. The computer data, which is what it is stored, and you can come back and look at that. So we don't do WGS as the first line testing in the NHS. But the very positive changes that have happened in the last 5 years in England is that the process of organising and delivering genetic testing has been formalised, with all laboratories established to offer that testing. The eligibility for testing is clarified within what's called the test directory, and a cardiologist with expertise or a geneticist can look at that directory. First of all, they can see whether the patient meets the eligibility criteria for testing, but they can also look at exactly what genes are on that panel. The process of adding in new genes when they're discovered is into those panels, so those panels are being reviewed. So there'll be a difference between 2022 and 2032. But the next question is the one that you've answered. Where does the patient fit into that? And there isn't a mechanism within the NHS for recalling and retesting. So what we advocate is to do that testing at the point of contact. So you have your annual appointment with your ICC specialist. They look at your file. We haven't got a gene identified. What do we do? And there is a decision made. Do we need to do a completely new test? Has somebody else in the family undergone testing recently? And we can look at that. And if necessary, if there's been a certain lag in time, probably 5 years plus, maybe it's time to do the testing. And we know that throughout England, tests have been sent to different laboratories, sometimes out of the country, and that is not encouraged. Now it is encouraged that all testing goes through the NHS laboratories.
Why is that, Ruth? Because I think there's an important point there, isn't there, about not all genetic testing is made equally.
Yeah. So it means that those 4 laboratories offer entirely standardised testing. So it doesn't matter if you're in Bristol or Sheffield, you get the same test and the same result. And that's really important because it's unhelpful if, one sample goes somewhere and gets one result. And you might imagine everybody would pick up the same, but because of the differences in technologies, that's not always, and differences in interpretation. And so it's about equity of access. This is now not a postcode lottery. This is, if you meet the eligibility criteria, you can be tested. Your cardiologist doesn't pay. The money goes to the laboratory. So again, if you meet the eligibility criteria, the test is funded. And it's about standardising the results that come out. It will be the same whether it goes to Sheffield or Bristol or London. Same report in roughly the same format, and that result can be used for testing in anybody within the family in that area.
If we think about the process for a patient, so a patient might present with symptoms, cardiomyopathy might be suspected, they'll go to a cardiologist, they'll have imaging done, clinically the cardiomyopathy will be confirmed. At that point, presumably the cardiologist will then go hunting for a gene. At what point would the cardiomyopathy patient come across your path, Ruth? At what point would somebody see a clinical geneticist as opposed to a cardiomyopathy doctor with a specialism?
That's a really good question, and I know that it does vary around the country, and to some extent that reflects the services that have been developed for cardiomyopathy. So because of the pressures on clinical genetic service in this part of the world, we have had to be quite restrictive over referrals. And the way we've progressed that is to work very hard with our cardiology colleagues in the process, and we call it mainstreaming. So encouraging them to do the testing rather than referring directly into the service. And consultants in clinical genetics work with genetic counsellors, genomic counsellors. And we, for example, here have designated genetic counsellors who work very closely with the cardiologists, and they do a lot of the testing. So we do distinguish between patients who have cardiomyopathy, where we are very comfortable with the cardiologist initiating testing, but where we recommend involvement of the genetics department is for family members who don't have cardiomyopathy, who are having testing in order to find out if they will develop cardiomyopathy. Some people call that predictive testing because at the moment you're okay and we're talking about something that might happen in the future. But many of those patients will actually be going through the cardiac evaluation as well. Yeah. ECG, sometimes more than that, imaging, as you've alluded to. I think there isn't perhaps there isn't an ideal. It should be tailored to the services you have, but also to the individual, because it is very different if this is a new finding in your family, nobody has had any information about this at all, then a genetic contact might be very, very worthwhile. But in some families, there's a lot of awareness around the condition, and a lot of the information that they seek can come from the cardiology team. I perhaps mentioned there's a completely separate pathway where the diagnosis arises from a sudden death. Where there is a pathway now through the coronial process that comes.
I was wondering about that because obviously, if the first you know about this sort of disease in your family is through a sudden cardiac death, and then you go down the sort of genetic digging process, is different, isn't it? And I think the other thing that interests me is the idea of a de novo mutation. So, i.e., the first time that the mutation has happened in the family. Can you talk to us a little bit about that? Because how do you find out that it's the first time it's happened in the family? Do you test everyone in the family? How does that process work?
Well, the reliable way is to test parents, and so that's often the first thing that we do. Obviously, in an adult age group, that's much more difficult. We can't always do that. So it's sometimes an assumption rather than proven. So if we can't test the parents, even if there's no suspicious family history, we can't rule out the possibility that's inherited genetics. So I think it's easier to rule it in than rule it out. So it's very hard to be certain that something hasn't been inherited if you can't do the testing. We also know that obviously biological relationships are not necessarily as stated. And also we do sometimes see what we call mosaicism, which is that this has arisen, there is a mild form, if you like, only some cells, and therefore you can see siblings who are affected when neither parent appears to be so. So in terms of practical advice for the families, it doesn't often matter whether it's de novo or not. The important thing is which gene it is and whether the pattern of inheritance is this dominant or recessive or, and that will be the mainstay of the information that's given.
I'm glad you mentioned patterns of inheritance. So I think cardiomyopathy is generally thought to be an autosomal dominant pattern of inheritance. Talk to us a little bit about that. Are they all that pattern of inheritance, and what does that actually mean?
No, not all. So there are more than 3 different patterns of inheritance.
I knew it wouldn't be simple.
It wouldn't be simple.
Yeah.
The common ones are dominant, and the prefix autosomal means that it applies to, so we have a set of chromosomes, 2 of which are sex chromosomes, and the others are called the autosomes. So we're talking about numbers 1 to 22.
Okay.
So autosomal dominant means that we have a pair of chromosomes, a pair of genes, and if we think of chromosomes a bit like strings of beads on a necklace, and each bead is a gene. We've got 2 copies of each gene, 1 from each parent. And a dominant gene shows this effect when one gene is affected, one gene has a variant. Whereas for recessive inheritance, both copies carry a variant. So the difference is in inheritance that for a dominant gene, there's a 50-50 chance that you pass on the normal or the not normal, the variant. For recessive, it's very different, and we have carriers and different risks. So the X chromosome, or sex-linked, which is rare, does feature in cardiomyopathy, and there are a number of genes that are X-linked, tends to be carried by females, and their sons are affected. But there are differences, and females can show signs, often less than the males, and that's to do with having 2 X's as opposed to 1 X and 1 Y, and the second X, if it's normal, can have some protective effect.
Okay.
So that is more complicated, and
even more complicated.
Even more complicated.
Even more, yeah. So I'm going to ask you, epigenetics, what is it, and is it important to the cardiomyopathy patient?
I think it's going to turn out to be very important to all diseases in explaining these unanswered questions about, why does something happen to somebody at the age of 5 and to somebody else at 55? And if that is your environment, it may be mediated through epigenetics. So for me, I guess my simple explanation of epigenetics is that you start with your DNA code and that you've inherited, but there's lots of processes by which that is active and inactive. So the code is read, and the way in which you read it is subject to all kinds of modifications, and we believe that some of those are environmental. And what are the effects of the environment? What's the effect of the environment within us, all the other genes, all the hormones, all the...
I was going to ask you about that. So when you say the environment, do you literally mean, the trees and the sea and the land, and whether we, live in a smoky environment or a hot environment, or do you mean a more sort of holistic way of talking about the environment? Both.
So very much both. So we do know already, about some of those internal environmental factors. How do other diseases affect cardiomyopathy? And then we're looking at external factors. So I guess think about alcohol as one of those. But yes, the wider environment. And these are very difficult studies to do. They're our capacity to measure those differences at the moment, I think, is very limited. But if you look at the progress that's been made in genetics and genomics in the last decade even, we have to assume that progress will continue. And I think for some of that is academic and of interest, and in a practical sense, I think it comes back to the questions about how does genetics help patients and patient care and understanding, and knowing what the triggers are. Why does the disease develop? Maybe that's where epigenetics will become helpful. What our responses to drugs are, which are the best treatments. So I think it does have practical value as well as being of interest.
My last question is really about the exciting new world of gene therapies. What are your thoughts around sort of gene therapies? You mentioned gene silencing. It opens up a world of possibilities, doesn't it, in terms of treatments? Maybe, I'm going to say this word out loud, okay, a cure for a cardiomyopathy. Is that on the horizon now, or is it still a little bit out of reach?
The definition of gene therapy, I think, is the understanding of what we mean by gene therapy is important. I think to most people, when we talk about gene therapy, they think there's a gene, it contains a variant or mutation, and what we're going to do is we're going to fix that. And that fixes, you then have a normal DNA code, and so that's that.
When you say fix, do you mean cut it out, essentially?
So if that is our model for gene therapy, we're going to fix the error, if you like. How do we do that? And there are studies that look at the ways of doing that, and gene editing is one of those. That's so what are the barriers to that? What makes that very difficult? Well, the most difficult thing is that you've got to do that in the cells, and you've got to do it before the process starts, probably. And for cardiomyopathy, is that just the cardiomyocytes? Where does that need to be, and where does that need to be delivered? And does that have any downside if you do that? What are the sort of side effects? So those challenges around gene therapy apply universally to all diseases. I think they're pretty much the same for cardiac as for any other organ. And I think it is inevitably going to be a big interest. How likely is it to yield results in the near future? I'd say quite slim.
Okay.
If you look at the number of gene therapy trials overall for all diseases and the numbers that have come to fruition to a meaningful therapy in the last decade, very, very small. And they tend to have been for very particular diseases where that model is likely to work, where, for example, you're trying to get something into the bloodstream, which is obviously easier than getting into heart muscle. I think when I give talks, when the question of gene therapy comes up, I tend to divert into how do we use genetics and genomics therapeutically, of which gene therapy is one therapeutic, and pharmacogenomics. And I think that's where I would say for the foreseeable future, the better developments will occur.
So by that you mean sort of medical therapy that is based on knowing the underlying gene?
Exactly.
Okay.
And there are already fantastic examples of pharmacogenomic interventions that prevent the development of a disease or treat, perhaps not to the point of cure, but very, very significant. And that's based on understanding the biology, understanding the pathway, and finding a drug. And sometimes these are newly developed drugs, but sometimes they're drugs that are already known that block the effect, for example, of the harmful gene. And I think that is much in terms of time scale, because the logistics of those studies are probably, and particularly for cardiomyopathy, I'd have thought that's going to be where we would see therapeutic gains using genetic and genomic information.
It's lovely to end on a positive note. Such a complex subject, which I think you've made eminently understandable. Thank you so much for taking the time to talk to me about it today. And as we were saying before, no such thing as a stupid question. I hope I haven't asked too many, but it's been a pleasure to talk to you, and thank you very, very much for coming onto the podcast.
You're very welcome. It is indeed a very, very complex area to summarise in a short time, and I think what's helpful is that, we can signpost people to other places where they can read more about the subject.
And you and I will go and have some lunch and a drink, shall we?
Absolutely.
Okay.
Thanks, Libby.
Thanks for listening to this episode of Cardiomyopathy Conversations. Please remember, any medical information contained within the podcast should not be used as a substitute for advice from your own doctors. If you have any concerns over your own condition, please do consult your medical team. Views expressed in the podcast do not necessarily represent the views of Cardiomyopathy UK, but are the views of the individual host or speaker and is intended for a UK audience. Cardiomyopathy UK does not accept responsibility for any actions taken after listening to this podcast.